And I'm going to go ahead and share. All right. So can you guys see the Capital Community College screen?
Yes.
Okay. So I'm sharing my screen. You guys can see the Blackboard screen right now?
Yes.
Okay, great. I don't know why this is acting different today. So the first thing I wanted to point out is just the syllabus. And here's our syllabus. I'm just going to scroll to the dates. So I have scheduled the first exam for that week, 9/21 to 9/27. It will probably be closer to 9/27 since today we're past 9/13 and we're just working on 11 and 12 today. Right, so all of this will get pushed a little bit. My point is I'm not going to have you take an exam when we haven't gone over all the information yet. I don't think that's fair. Okay, so that's the first thing I wanted to point out. Did any of you that are on the call, did you have trouble uploading the homework assignments?
I did not.
Well, I didn't have issue. I just I wasn't unaware when it was due.
Okay. So I'm trying to make the homework due, trying to give you a week basically to do it. My reason for that is just that we're going over 2 more chapters today. And so, then all of a sudden after today, if you have to do homework for 4 chapters, that's kind of a lot and it gets overwhelming quickly. My goal with the homework is to make you guys essentially create your own outlines for the chapters to help you organize the thoughts for the chapters, which will help you study for the test. So I do want to, grade those homeworks tonight. I'm gonna work on hopefully all of them, but grade them and give you guys feedback so that you can use those as study guides for your test. And then what I will assign today and tonight probably I will create homework assignments for chapters 11 and 12, similar things where I'll have you outline some things and tell me these 3 things or those 3 things, and it just kind of helps you, I think, to organize your thoughts or helps some people to organize their thoughts. So in addition to giving you homework points, it will serve as a study guide for the test. So if you haven't handed them in and you can still hand them in, I'm pretty sure Blackboard allows you to do late submissions. It just shows me when you submitted it. So go ahead and do it. I will still give you credit for it. So it's much better to do it than to not do anything. Not only will you get points, but I'll give you some feedback on what you handed in so that it will help you study for the test. Today, what I want to do is go over chapters 11 and 12, which are, you can see that they were in the PowerPoint presentations folder and the folder labeled week 2. Okay, I've already opened this up. So can you that are on the call, can you see this? Can you see this PowerPoint?
Yes.
Yes, okay, good. So chapter 11 is all about prokaryotes, chapter 12 is all about eukaryotes. So you should already know from last week the difference between prokaryotes and eukaryotes, or you should start to have an idea about that. Prokaryotes are simpler, one-celled organisms. They are simpler cells, but there tends to be more of them that can be pathogenic to humans. So we study them a little bit more. Eukaryotes will be things that are a little more multicellular. We'll study some fungi and we'll get into some of the characteristics of those. Okay. So the first thing that they talk about as the morphology of prokaryotic cells. So who can tell me what morphology is? That word means. Anybody?
What they look like, their shape and stuff?
What they look like in their shape. Yeah, exactly. That's exactly what it is. So many of these things, many of these prokaryotic organisms are just organized by what they look like, right? Which made sense. If you don't have much else information, much more information on them, you could at least look at them under a microscope, and things that look similar, you group in similar classes, right? That makes sense. So here are just some of the different morphologies. Coccus would be a ball like that. Coccobacillus would be somewhere between a ball and a rod shape. Bacillus is always a rod shape, and and on. You can go into the different shapes of the different bacteria. Some bacteria produce endospores. Bacteria that produce endospores tend to be gram-positive. So you can see those two types, Bacillus and Clostridium, produce endospores. And an endospore is a way that a bacteria can kind of go dormant for a while, right? So bacteria are living organisms. They need food. They need energy to live. When food is really short in supply, they can actually germinate. They can make these endospores, right? And they're essentially just going dormant so that they can last for several years without any food, without any nutrition sources. And then when there is food available, the endospore germinates to form what's called a vegetative cell, which just means it's a cell that's actually alive and eating and functioning the way it should. So you can see that those things that produce endospores are actually very hard to kill, right? The ones that can produce endospores and cause pathogenic diseases to us are very hard to get rid of. So they're a big concern to people who process food, people in healthcare environments, right? Those are the ones that you need to eradicate that give you more problems. You can't just starve them out necessarily. There are three main methods of reproduction for these prokaryotes. There is binary fission, snapping division, and budding. Binary fission is the most common. So binary fission is just where one cell grows bigger and bigger and bigger, doubles everything inside of it, and it splits into two new cells. That's the easiest way to think about binary fission. Snapping division occurs in some gram-positive bacilli, and we'll talk about that later. And then some prokaryotes divide by budding. You can see an example of budding over here on the right on the slide. The DNA, which is attached to the cytoplasmic membrane, is replicated first, and then it kind of elongates, and you get this bud coming off of the cell, and that bud just eventually breaks off and becomes its own cell. Not quite the same as binary fission, where it just splits down the middle. This kind of buds off the side. The arrangements result from two aspects of division during binary fission. So during binary fission, which is the way most of these cells reproduce, again, they double everything inside, they get bigger, then they split into two new cells. And so as they're splitting, they look like these cells that are linked together. Right? So the arrangements are very specific. The way that they split is very specific. So you can see that over here on the left, you've got a cocci, right? It's one of those balls. And you've got diplococci, which means you've got doublets of them. If you talk about something that's streptococci, it's a string. Strepto is a string of them. You've got tetrads, you've got sarcinae, and staphylococci. So a lot of bacteria are named based on the way they divide and the way they look when they divide. You guys, I'm sure, have heard of strep and have heard of staph, right? It's because that particular strain of that bacteria divides in a certain way, and that's what it's named for. Okay, and so this on the left is showing cocci, the small round bacteria. The many bacteria are that rod-shaped, and those are called bacillus. And so again, you've got diplobacilli, streptobacilli, right? A string of bacillus bacteria, here they are, which streptococci is a string of the little brown bacteria, palisades and V-shape. So a lot of times the bacteria are named for the way that they actually divide, the way they look when they divide. Again, a lot of these things are named by the way they look because that's the most information people had about them for a long period of time was you just knew what they looked like under a microscope, so that's how you named them. So this picture can be kind of overwhelming at first. It's the modern classification of bacteria. So what that means is bacteria were always classified based on how they looked. But now that modern techniques are available and DNA sequencing is available and all that sort of thing, there's more information that scientists have about these bacteria. And so they group them a little bit differently. And they group them, they can be a little more specific in how they group them. And so what they've created are 24 groups called 24 phyla. Okay. So you've got a big group is called a kingdom, and within a kingdom you have phyla, right? Each single phyla is called a phylum. So a phylum is just a group, a smaller group within a kingdom. So in this big group of bacteria, you've got 24 of these smaller groups. And those groupings are based on the sequence of their 16S rRNA. Now, this goes into ribosomes, okay? So in general, what are ribosomes? What do they do in the cell? Who can tell me what they do?
Transport?
Not transport. Good try. They actually are the protein factories. They're where the proteins are made. Okay, so ribosomes are responsible for making proteins. And rRNA, that little r before the RNA, stands for ribosomal RNA. And so it's actually the sequence that encodes for the ribosome. These phyla, these groupings of bacteria, are based on how similar their ribosomal RNA is, how similar their protein factories are. And so you can see that, for instance, these groups that are over here on the left are all kind of clustered together. Well, they have very similar protein factories, right? Those gram-negative bacteria have different protein factories than, say, the archaea, which are over here on the bottom right. Okay, and that's what these groupings are based on. And so it turns out that a lot of these groupings fall in line with the groupings that were already established, right? It's not like this is a totally different thing from the way they were grouped before. It turns out that you can see there's kind of clusters here. There's gram-negative bacteria over here on the left bottom corner. There's gram-positive bacteria. You've got some low GC content gram-positive bacteria clustered together. You've got archaea on the lower right. Okay, so it's just showing where these bacteria are based on their sequence of ribosomal RNA. So some of these groups, they're describing some of these groups. Some of these groups are called deeply branching or phototropic bacteria. Deeply branching bacteria, scientists believe that these organisms are similar to the earliest bacteria. So let me flip back. So deeply branching bacteria are actually listed up here. The earliest bacteria are probably the archaea. Okay, but you can see that there are things like thermophilic bacteria, green non-sulfur bacteria. We'll get into these in a second. So they're really old, right? They were some of the first life forms to develop. They are autotrophic. Who can tell me what autotrophic means? Anybody?
They can like produce their own food using light or water, CO2.
Absolutely. They can produce their own food. So auto, right? Auto is something they can do automatically. Autotrophic means you can produce your own food. Some organisms are considered heterotrophic, like us, like humans, like people, or like animals. we can't produce our own food, right? We have to eat things. But plants are autotrophic. They can produce their own food from the sunlight. So some bacteria are autotrophic. This group of deeply branching bacteria, they are all autotrophic. They live in habitats similar to those thought to exist on early Earth, which means early Earth, there wasn't a lot of oxygen around, there was a lot of water around. So these are living in habitats which are maybe not what we're used to now, but are probably what Earth was like a few million years ago. Um, and here are some of the ones that fall into that category. Aquifex, which is considered to be one of the earliest branches of bacteria, and Deinococcus, which has an outer membrane similar to gram-negatives, but it stains gram-positive. So that's unique about that particular bacteria. Another group are phototrophic bacteria. So if autotrophic means you can make your own food, what do you think phototrophic means? What's photo? What's that prefix?
Has to do with light.
Has to do with light. Excellent. So phototrophic bacteria make their own food by light, by using light. So the way plants do this, right? The same way that plants do this. So you can see that this group of phototrophic bacteria is divided into 5 smaller groups based on what pigment they use. So using a different pigment, it's going to absorb a different wavelength of light. So you want, a different light source depending on what the pigment is. So you can see these 5 groups that are in there are just based on the colors, which is based on the pigments again that they each have. So here are the characteristics of those major groups of phototrophic bacteria, right? So we had 5 of them here. Here are the 5 of them. So even though we have the common names, and that's what's listed on this previous page, they do all have class names, which are more scientific names. Okay, so you need to know the class names. You need to know cyanobacteria are the blue-green bacteria, or sometimes called blue-green algae. Okay, you can see that they all have different photosynthetic pigments, right? So they undergo different types of photosynthesis. Now, what do you think oxygenic is versus anoxygenic?
Is that same as anaerobic, anaerobic?
Correct. So oxygenic uses oxygen, anoxygenic does not use oxygen. It gives you the electron donor, which is at the end of the biochemistry of photosynthesis. Whether or not sulfur is deposited, uh, nitrogen fixation, and motility. Okay, so this is a very nice summary. This table is a nice summary of those 5 major groups of phototrophic bacteria. You will also see when bacteria is listed and grouped together, sometimes it's grouped based on the G plus C content. So what are the 4 letters of DNA, the 4 nucleotides that are used? Who can tell me that? They may know what DNA is made of.
I
Um,
did it. Oh, go ahead.
I just know like the letters are A, T, and G, C.
Is
Good,
it
perfect.
guanine, thymine, adenosine, and I can't remember the last one.
Thymine. Yep,
Thymine. Yes.
good, good. So bacteria are living organisms, right? And they have DNA just like we have. So some bacteria are classified based on the amount of G's and C's that they have. That's all it is. So it's the percentage of all base pairs in a genome that are guanine and cytosine base pairs, G to C base pairs. And people who classify bacteria use that as a characteristic to classify bacteria, particularly those that stain gram positive, right? So you have all bacteria, you do a gram stain test on them. Some are gram positive, some are gram negative. Of the gram positive ones, you can do a test to see if the GC ratio is above 50%, if there's a lot of GCs, or below 50%. And you classify those, you split that group of gram-positive bacteria into those two groups based on how many G's and C's they have. That's all it is. Okay. There are some that are low GC gram-positive bacteria. One of particular importance to us is Clostridia. It is a rod-shaped organism. It is an obligate anaerobe. It's important in medicine and industry. It's related to Eupolpicium, which is a sulfate-reducing microbe, and Selenomonas. So it's an obligate anaerobe. We need to talk about that. What is anaerobe?
Can survive without oxygen.
Good. So the fact that it's an obligate anaerobe means it only survives without oxygen. It means if you put enough oxygen around it, you will kill off the Clostridia. It only will survive without oxygen. Obligate anaerobe. The other term that you might see is a facultative anaerobe. And what that means is it can survive without oxygen if it needs to. But an obligate anaerobe means it's obligated to be anaerobic, right? It can't survive with oxygen. It has to survive without oxygen. And so you'll see that some of these bacteria are classified as anaerobic, some are aerobic, some are obligate anaerobes, right? Some are facultative anaerobes. Another one of the low GC gram-positive bacteria would be the mycoplasmas. They can be facultative or obligate anaerobes. So some species cannot survive with oxygen around. Some species just would rather not have oxygen around. They don't have cell walls. Okay, they're the smallest free-living cells, and they colonize mucous membranes of the respiratory and urinary tract, so they can cause infections. And a lot of these I'm going to go through rather quickly just because it's a matter of just memorizing the different groupings. I'm just trying to give you reasons why they're grouped together. Hopefully that'll help you with the remembering it. Other low GC gram-positive, bacilli and cocci. Bacillus is common in many soils. Bacillus thuringiensis is used by farmers and gardeners as an insecticide. Bacillus anthracis causes anthrax. You can see the bacillus. Here are the bacteria in this picture here. You can see the toxins that they make. So the toxins are what causes a disease to us. Other low GC gram-positive bacilli and cocci, Listeria and Lactobacillus. Listeria can contaminate milk and meat products. It is capable of reproducing even under refrigeration. And it's one of the bacteria that can cross the placenta in pregnant women. So it's one that if you're pregnant, you have to be careful around things like dairy and meat. As long as they're, you have to just make sure that they're kept in conditions where they're clean. Lactobacillus grows in the body, but it rarely causes diseases. It's used in the production of a lot of foods, so it actually is something that will be used to cause foods to be sour or fermented, used for yogurts and things like that. Other ones, Streptococcus and Enterococcus, cause a lot of diseases. There are a lot of these that are drug resistant, and Staphylococcus is one of the most common inhabitants of humans. This produces toxins and enzymes that contribute to disease. So these are big players in diseases like in hospitals and nursing homes, stuff like that. You want to make sure that you're looking out for things like Streptococcus and Staphylococcus. They cause really nasty infections, and many of them are drug resistant now. So again, here are the bacteria we've been looking at. They've got your GC percentage low, right? Here are the genera, which just is a smaller grouping. So within this phylum and class, there are these three groups, which are called genera. It's the word for genus, right? And here are the special characteristics of each genera and the diseases they can cause. So Clostridium was the one that forms endospores, causes tetanus and botulism. You can, I used to teach dental hygiene students. So they were, worried about, not worried, but they learned a lot about Selenomonas, which causes dental caries, which are cavities. So you can see what some of these different things cause. Bacillus can cause anthrax. Lactobacillus can cause blood infections, but not very, very many. Streptococcus obviously causes strep throat and scarlet fever. Staphylococcus can lead to food poisoning or like skin poisoning. If you have a staph infection, sometimes that's like when your skin gets really infected. So those were the low GC gram positives. We have high GC gram positives. Corynebacterium, Mycobacterium, Actinomycetes. So again, the first group here, Corynebacterium, they are pleomorphic aerobes and facultative anaerobes. What that means is pleomorphic means they can change shape, right? Morphism is the shape. So with oxygen, they can change shape and function. But they can survive without oxygen. They're facultative anaerobes, which means that they can always find a way to survive without oxygen. So you can't just deprive them of oxygen and hope that that will kill them off, because they can survive without oxygen. Okay. Mycobacterium, those are aerobic rods that can form filaments. They have a slow growth because of mycolic acid in their walls. So these need oxygen. Okay. And Actinomycetes, this is a similar word to a fungus, and it's because it forms branching filaments that look like fungi. So this group contains several genera that are of particular importance if you're studying disease. So here's a nice summary of the GC, high content GC bacteria. However, gram-negative bacteria are a much larger group, a much more robust group. They are the largest and most diverse group, so you can think about them in almost every environment. There are 6 classes of these proteobacteria, and if you look at the names, they're just alpha, beta, gamma, delta, epsilon, zeta, right? They're just Greek letters. So they're just named like the first group, the second group, the third group. The first group, alpha proteobacteria, are nitrogen fixers, so they grow with the roots of plants. They're really important to agriculture. So if you do any, farming or planting or gardening, you know that nitrogen is really important for the soil, and a lot of fertilizers have nitrogen in them. But the nitrogen has to be in a particular form for the plants to take it up. And in order for it to get to that form, you have to have bacteria associated with the roots that are in the ground, and that bacteria transforms the nitrogen to a form that the plant can then take up. And these are groups of bacteria that can do that. That's what nitrogen fixers means. It means that they can take nitrogen in one form and put it into another form so that the plant can actually take that up. Okay, so here are just some examples of these alpha proteobacteria, what they do. Then we get to a class called beta proteobacteria. These are some pathogenic ones. You can see Neisseria can cause numerous diseases. It inhabits mucous membranes. Bordetella causes pertussis, whooping cough. That's if you have animals, like especially dogs, when they stay at a kennel, they have to get vaccines against Bordetella. Burkholderia colonizes moist environmental surfaces, so like respiratory passages. People with cystic fibrosis obviously don't have the ability to fight that off as much as most people do. Here's some other beta proteobacteria. Just trying to go through and see if there's any words. Flocks. Flock means what you think it would mean. It's a group of them. So if you get a flock of Spiracillus, it will impede flow of water or of waste in treatment plants. So a flock is just a group of bacteria that are impeding that flow. The third group, gamma proteobacteria, it's the largest and most diverse class of these 6. It's divided into subgroups because it's so diverse. So here are the subgroups listed out for you. Right? And then it goes through what some of these are. There are some that are intracellular pathogens, which means they can infect your cells. There are some that use methane as a carbon and energy source, meaning they can live without oxygen. They inhabit anaerobic environments. So again, you would think with living things, you can take away their oxygen and they can't live, but there are plenty of bacteria that can live without oxygen. There are glycolytic facultative anaerobes. Again, this facultative anaerobes means they can live without oxygen. Those break down carbohydrates by glycolysis and the pentose phosphate pathway, which if you go into biochemistry, you'll learn all these different pathways of how you break down a sugar. Pseudomonads can break down a lot of different organic compounds. They're an important pathogen for humans and animals. They can cause UTIs, ear infections, lung infections, right? There's some examples of those. So far, right, we've got Enterobacteria, Vibrio, Pastoracea. These are all, here are the genera that are represented, right? Here are the typical human diseases. So these are all gram-negative bacteria that fall into the group of gamma proteobacteria. Then we get to the class epsilon proteobacteria and some important genera, Campylobacter, Helicobacter, okay? And that's about it that I'm going to say about the prokaryotes. So one of the things that I don't love about teaching microbiology is that it's kind of dry, right? It's like, oh, there's this group and here's what they do, and there's this group and here's what they do. Charts are always a good way to remember some of these things. And again, I'm just trying to give you reasons why they're grouped together. So a lot of times if you draw out a like a big flow chart, right, you can say, okay, here's my big group of all bacteria. And then I'm dividing it into gram-negative and gram-positive. And then I'm dividing the gram-positive further into high GC content, low GC content. And that's where these fall, right? And then I'm going to gram-negative bacteria and I'm looking at the proteobacteria that are gram-negative and I'm going to divide those into 6 groups, alpha, beta, gamma, epsilon, and so forth. So I think that's the kind of thing that you need to do to be able to study this chapter. Are there questions on some of these prokaryotes that we went through?
You want us to be able to be more than pretty familiar. Like, for example, would you give us a question that says A, B, C, D, this type of bacteria is facultative anaerobic, certain this shape and that shape, and we'd have to identify it from A, B, C, or D?
Yes.
Okay, thank you.
Sorry, I hate that. I hate those kind of questions, but yes, that's kind of what microbiology does. Yeah, so knowing the groups is really important. Knowing, like you said, whether it's a facultative anaerobe or an obligate anaerobe, knowing which ones make endospores is really important. And again, there's a lot more bacteria than the ones that we went through. I just went through some of the ones that are important either because they can cause disease or because they're used in food production, right? So these are bacteria that the ones that I've gone through are the ones that are more important for things like that. So I do take questions from a test bank, but I read through the test bank first, and I only select the questions that are bacteria that we named. You know what I mean? So I'm not going to just blindly take any questions from the test bank because I don't feel that that's fair. So I do go through the test bank and sort out the questions. Chapter 12 is doing the same thing, only with eukaryotes. So characterizing them and classifying them, basically same type of thing. These groups were created based on their characteristics, right? They look similar, or they, obtain food in a similar way, or they do something similar to another type of organism, and so they're kind of grouped together. Okay. This is microbiology, right? So we're talking about microorganisms. We tend to think of microorganisms only as bacteria, and we do study viruses in here, even though they're not alive. But there are microorganisms that are eukaryotic. They're a little more complex. There are protozoa. There are fungi. There are algae. There are slime molds and water molds. Some of these are human pathogens, and some of them are just vital for our life just because of things they do in the environment around us or things that we benefit from, such as food production or medicine production. Okay, so reproduction, the nucleus has one or two complete copies of the genome. If it has a single copy, it's called haploid. If it has two copies, it's called diploid. So most fungi and a lot of algae and a lot of protozoa, right? They live in a state where they only have a single copy of the genome. So the genome that's all the genes that are known to your particular species, right? So for humans, we have two copies of the genome. We have 46 chromosomes in every cell, and we get 23 chromosomes from our mother and 23 from our father, right? So that's where the two copies come from. So we are called diploid because we have two of each chromosome. And all animals exist as diploid organisms. All plants exist as diploid organisms. Some fungi, right? And if you remember, I kind of said, I think last week, that protozoa reminds me of the junk drawer, right? So in eukaryotes, you have animals, plants, fungi. They're kind of the higher order things. And then you have this class called protozoa, and algae kind of fall into that protozoa class. And the protozoa, some of them act like plants, some of them act like fungi, some of them act more like bacteria. Some of them are single-celled, even though they're these eukaryotic cells. So they do funky stuff, okay? There's two types of nuclear division. So there's two types of division of all the DNA that's in the nucleus. There's mitosis and meiosis that goes on. Mitosis is just the same as binary fission. The cell makes a copy of everything, gets bigger and bigger and bigger. When it has two copies of everything, it splits into two new cells. Meiosis is different. Meiosis is how we as animals make sperm and egg. And it's a different type of division where the end result is that the cell at the end only has one copy of all of your genome. So at the end of meiosis, a sperm cell has 23 chromosomes. And at the end of meiosis, an egg cell has 23 chromosomes. And it's only when the sperm fertilizes the egg that you end up with a cell with 46 chromosomes that can grow into a new human being. Okay, so those are the two types of division. So one of the major differences between prokaryotes and eukaryotes, prokaryotes only divide asexually, right? They only split up their DNA asexually. Most of them split into two new cells, or they bud into a new cell. A lot of these organisms actually have more of the sexual reproduction process, where you have one organism making cells that have 23 chromosomes, and you have a second organism making cells that have 23 chromosomes. And those two cells come together to make an organism with 46 chromosomes. Okay, so that's true of animals, of plants, of a lot of the fungi, and of some algae and protozoa. So classification. So early groups group these organisms based on structural similarities. So what they look like, right? Things that looked similar got grouped together. But the modern classification is based more on similarities in nucleotide sequences. So as I said, with prokaryotes, technology has come far enough that we can get the DNA sequences of a lot of these things. And scientists use that more to classify these organisms. And in doing that, some organisms have been kind of moved around in their classification. But most of them haven't. Right? Most of the time, if their DNA is similar, they're going to look similar and behave similarly, right? That's just the way it goes. So right here, it talks about protists. Again, protists is the junk drawer. It's an informal grouping of eukaryotes. They're not plants or animals or fungi. Algae and protozoa are both in the grouping called protists. Protists are, like I said, they don't fall into the grouping of plants or animals or fungi for one reason or another. They usually act a little bit like one or multiple things. They usually act a little like plants and a little like animals, or a little like fungi and a little like plants. So they get grouped in this their own category. And so here are just some pictures of different protists. Right. So changing classification over the centuries, right? We don't need to worry about this too much, but Linnaeus, right, had plants, animals, right, on this big timeline. And then late in the 20th century, they kind of figured out that a lot of what they were calling plants were really fungi. A lot of what they were calling animals are really protists, right? Plants and animals stayed the same. And then as time went on, early 21st century, now we've got so much more sequencing going on. Fungi are fungi, plants are plants, animals are animals. Look at all these different types of protists that we have in there, right? And that's a lot of these are actually what you're studying in microbiology. These different protists are just in different groups based on their sequence, their DNA sequences. So protozoa fall into that group of protists. They are a very diverse group. Like I said, I think of them as a junk drawer. They've got a lot of stuff going on. They are all made of eukaryotic cells. So even if they're one celled, which they are, they're all unicellular, they're all eukaryotic. Previously, all of the cells that we talked about in the last chapter were prokaryotic and unicellular, right? So these are the only ones that are unicellular and eukaryotic. And they all lack a cell wall. So we'll see that a lot of bacteria have a cell wall. And we saw that last week. None of these protozoa have a cell wall. Most of them are motile. Most of them can move around. They have these little extensions. They have cilia or flagella, which are little kind of hair-like projections that can flap back and forth, push the thing around through the liquid. Some of them have pseudopods. If you've ever seen an amoeba move, it kind of has these changes shape, so it kind of has these feet that pull it along. Those are what pseudopods are. There is one subgroup called apicomplexans that can't move. That's the only group of protozoa that can't move. As far as where they live, they require water, right? They need to be near water. They're considered a very early group of organisms, so they haven't adapted real well to dry land. Most live in ponds or streams or lakes, oceans. Others lived in moist soil, beach sand, which is near the water, gets some water, or decaying organic matter, which has liquid in it or has water in it. Very few of them are pathogens, so very few of them can cause disease in us. So here's the morphology, just what they look like, right? So we're studying these in microbiology because they are microscopic, even though a lot of them don't cause disease. They have a lot of different morphologies. There's a lot of different types that look different. Some have two nuclei, a big one and a small one. There are different kinds of mitochondria. Some have a lot of mitochondria. What do mitochondria do in the cell?
Create energy, provide
Create
energy.
energy. Good, create energy. So some cells have a lot of mitochondria. Some of these protozoa have a lot of mitochondria, which would, hint at the fact that they maybe move around a lot. Others don't. Some have contractile vacuoles that pump water out of the cells. That's similar to a plant cell. Plant cells have contractile vacuoles for that very reason. They have different stages in their life cycle. Some of them move around when they're in a feeding stage called a trophocyte. Some of them have a resting stage called the cyst, where they actually look different. Which stage they're in makes them look different. Nutrition, how do they get their energy? Most are heterotrophic. So I told you in the last chapter that heterotrophic means they have to get their food, right? Autotrophic means they can make their food. What is chemo? What's that prefix mean? Anybody know?
Does it have to do with chemical?
Chemical, exactly. So chemotherapy is a chemical therapy, right? So most of these are chemoheterotrophs, meaning that they have to obtain their food by eating essentially some sort of chemicals. So some obtain nutrients by eating bacteria or decaying organic matter or other protozoa or some kind of host tissue. They have to eat something. They have to get energy from somewhere, and they can't make it themselves. There are a few that absorb nutrients from the surrounding water. Remember, a lot of these live in water. And then there are a couple specific types, dinoflagellates and euglenoids, that are photoautotrophic. Now, what does that mean, photoautotrophic?
They make their own energy with light?
With light, absolutely. So those two groups, dinoflagellates and euglenoids, actually can undergo photosynthesis, just like a plant cell can. So now you're getting to see how this is kind of just a catch-all of all these different organisms. Reproduction, most reproduce asexually by binary fission, so they just grow and double everything in size and then split into two. But there are a few that have sexual reproduction. Um, some become gametocytes that fuse to form diploid zygotes. So some of them would have just one set of chromosomes, and then you have another cell with just one set of chromosomes, and those two cells fuse together to have two sets of chromosomes. Um, some utilize a process called conjugation, which we'll see later on, where one cell can actually connect to another cell and just share DNA with it. It's kind of weird, but they can do that. Classification. So it has shifted over the years. It's constantly being revised and updated based on DNA sequencing. The current scheme, and this is from the most current book, groups protozoa into 6 groups, and there they are. Okay. So then it just goes through the 6 groups. This first one, Parabasala, lack mitochondria. So they don't have any of these organelles that can produce energy. But they have a nucleus. They don't have 2 nuclei, just one. They contain something that looks like a Golgi body. And important ones are Trichonympha and Trichomonas. The Diplomonadida also lack mitochondria. Okay, they also lack Golgi bodies and peroxisomes. They have 2 nuclei and multiple flagella, so they can move around. Giardia is kind of the prominent one that you will study. Euglenozoa have characteristics of both plants and animals. These are ones that can actually undergo photosynthesis, right? So they're photoautotrophs. They do have a flagella, which means they can move around, which that's more like an animal because plants don't move around. They do have mitochondria, so we're getting to something that's a little more complex. It's got a mitochondria in it. And there are 2 groups. There's euglenids and kinetoplasts. So this just goes into those 2 groups a little bit further. Euglenids are the ones that are photoautotrophic. So here's a drawing of one. They actually have chloroplasts in them, so they can take the sun's energy and make sugar from it. Kinetoplastids have a region of mitochondrial DNA called the kinetoplast, and some of these are pathogenic. So within Euglenozoa, there are euglenids, which are photoautotrophic. Kinetoplastids are not photoautotrophic. They have to get their food somehow. Okay, then we have a group called alveolates. They have membrane-bound cavities called alveoli, but nobody knows what they do, really. They are divided into 3 subgroups. So they're divided further into ciliates, apicomplexans, and dinoflagellates. Ciliates, as their name implies, have a lot of cilia to move themselves around. They are chemoheterotrophs and have 2 nuclei. There's only one that's known to be a pathogen to humans. Apicomplexans are also chemoheterotrophic. They're a pathogen for animals. They have a lot of organelles, which allow them to penetrate host cells. So things like Plasmodium, Cryptosporidium, Toxoplasma cause disease in humans. Plasmodium can lead to malaria, right? That gets in your blood. Toxoplasma is one that's found in cats, like litter boxes. Like they say, if a woman's pregnant, she shouldn't change the litter box because this particular toxoplasma, this particular protozoa, is found in litter boxes. Another group of these protozoa are dinoflagellates. Again, they're unicellular. All these things so far have been unicellular. They do have photosynthetic pigments. Historically, they were thought to be an algae, but now that we have more information because of DNA sequencing, they're classified as protozoa. A lot of these make up the plankton layer in freshwater and saltwater. If they're mobile, they have two flagella, which is weird. It's what this diagram is over on the right. They have one that's a tail and one that kind of wraps around the middle of it. So they spin around kind of like a top. When you get an abundance of these in marine water, that can lead to red tides. Okay. So some of these do produce neurotoxins, right? When there's red tides, they ban you from going in the ocean. They say you shouldn't go in because you can get diseases from it. Then there are rhizaria. These are amoeba that move and they can feed with pseudopods, so they kind of move around. They don't look a lot like anything else. Rhizaria are amoeba with thread-like pseudopods, and there are 2 groups within Rhizaria, Foraminifera and Radiolaria. Okay. And then there are amoebozoa, which are different amoeba. They have the lobe-shaped pseudopods and no shells. And this group includes Naegleria, Acanthamoeba, and Entamoeba. Slime molds, which used to be fungus, are now classified as an amoeba. There's plasmodial slime molds and cellular slime molds. So again, here are the different categories and distinguishing features, right? And what you see in bold, there are 4 on this page and 2 more on the next page. Those are the 6 groups within protozoa. And then the groups that are not bolded are within that bold group. So in other words, euglenozoa is subdivided into euglenids and kinetoplastids. Alveolates is subdivided into these 3 groups. Okay. So again, I always find it helpful to make a flow chart, right? There's protozoa, which you divide into 6 groups, which then you can divide into further groups depending on where they are. Questions on protozoa? And there's just a little bit more to get through on eukaryotes. Fungi. So protozoa are the hardest ones to get through because there's not a lot of uniformity between them. All fungi are chemoheterotrophs. So they all need a food source. None of them can make food. None of them can do photosynthesis, right? They do have cell walls. Usually the cell walls are composed of a material called chitin. They do not perform photosynthesis. They don't have chlorophyll. They're not plants. They are more closely related to animals. Significance of fungi, right? They are decomposers. So they actually decompose a lot of dead organisms and recycle those nutrients back to the ground, to the earth, which they help plants absorb water and minerals. They can do what's called nitrogen fixation too. A lot of them are used for food or in religious ceremonies, in the manufacture of food and beverage, especially anything that's fermented. A lot of them are used to produce antibiotics and other drugs. pharmaceutical industry is largely based on fungi. They can serve as important research tools. Again, think of pharmaceuticals. 30% cause disease of plants, animals, and humans. So some of them are pathogenic, and they can spoil fruit, right? Pickles, jams, and jellies. The morphology, there's two basic body shapes. There's molds and yeasts. Molds have these long filaments called hyphae, which you can see in the upper right of the slide. Yeasts are small globular things which are composed of a single cell, but they live as multiple cells together. So each one of these little round things is a yeast. Some fungi are dimorphic, meaning that they can produce both of these different shapes, right? So at some point during their life, they are hyphae shaped, and at some point during their life, you have yeast-like shapes. And they can change based on the environmental conditions, right? So whatever is going on in the environment, they change to what's best for that condition. They have to acquire nutrients. They can't make them. They do this by absorption. Some can trap and kill microscopic soil-dwelling nematodes, not very many. Haustoria allow for some fungi to derive nutrients from living plants and animals. Most of them are aerobic, so most of them need oxygen around. However, many yeasts are facultative anaerobes, which means that a lot of yeasts can survive without oxygen. Reproduction, they all have some means of asexual reproduction, which involves mitosis and the cell splitting, but they also most reproduce sexually. So they have a very complex life cycle that involves one part of their life cycle where they split, one cell splits into two new cells and they're exactly the same. And then that cell can actually split into cells that are half as big. And then those cells that are half as big come together to form another full cell again. So definitely look over the reproductive cycle of fungi, right? This is something that I'll ask you to kind of outline a little bit on the homework because it's something to definitely get into a little bit. Okay, and so here is actually the life cycle of it. So you can see there's a haploid stage and a diploid stage. Haploid means it only has one set of chromosomes, and diploid means it has two sets of chromosomes. So this is something that I'll have you guys review on the homework because I think it is really important. Then we get into classification. There's 4 different classifications of fungi. There's Zygomycota, Ascomycota, Basidiomycota, and Deuteromycetes. Okay. So in Zygomycota, there's 11,000 known species. Some are parasites of insects and other fungi. They reproduce asexually, and there are microsporidia, which used to be classified as protozoa, but now they're classified as fungi. Genetic analysis made scientists move them. These are obligate intracellular parasites, meaning that they have to live inside the cell of another species. So these are small resistant spores that live inside host cells. There is Ascomycota, right? And if you thought there were a lot of Zygomycota, there's 32,000 known species of Ascomycota. They form ascospores in sacs called asci. They also reproduce by conidiospores. They include most of the fungi that spoil food. So if you think of fungi that grow on, bread or anything else that you can think that you leave out that might spoil. Some can infect plants and humans. Athlete's foot is classified as an ascomycota, but many of these are beneficial, right? We use them to grow up a lot of the medicines that we use, like penicillium, Saccharomyces. And then there are Basidiomycota. These are mushrooms, right? So I think we can all picture what a mushroom looks like. Mushrooms are called basidiocarps. It's only actually part of the fungus. If you've ever studied fungus before, the mushroom that you see above the ground is only a small part of the actual fungus. There's a huge network underneath the ground of basically a fungus growing. so that it can take up all these nutrients from the ground. So what you see above the ground is a really small part of it. Most of these are decomposers. They decompose things that are dead, and then they return the nutrients back to the soil. Many of the mushrooms produce toxins or hallucinatory chemicals, and there's a couple of them. There is a fungus that's in this class that causes meningitis, and many of them cause expensive crop damage, right? They end up kind of taking over easily. And then the fourth group is deuteromycetes. This is a collection of fungi with unknown sexual stages. Most of them belong to the division Ascomycota based on their ribosomal RNA analysis, but it's no longer considered a formal taxon. So really these used to be their own group, their own division, and they're not anymore. They're pretty much just put under Ascomycota now. Then we get to lichens. Lichens are a partnership between fungi and photosynthetic microbes. So it's a symbiotic relationship. Does anybody know what a symbiotic relationship is?
They rely on one another.
Good, they rely on one another. So you have a fungus and usually an algae that rely on each other. They're abundant everywhere throughout the world. They grow in almost every habitat. And they're eaten by many animals. They're a food source for a lot of things. But basically, the fungus protects the algae, right? Physically protects the algae, right? It keeps it in a certain place. And the algae is photosynthetic, and so it's producing sugar that the fungus needs to live off of. That's the symbiotic relationship between them. And you've probably all seen algae growing on the side of a tree, or growing on a rock, or growing on a roof of a house. Lichens growing on all these things. So here is the makeup of a lichen. And again, here is the three divisions of fungus and their distinguishing features and the genera. So, are there questions about the stuff that we talked about today? You guys still there?
I'm here.
Okay. I know, it's kind of long. Okay, so are there any questions about the things that we discussed today? I mean, it's a lot of it is like going back through, like I said, creating flow charts, saying, okay, these live in this division, these live in this division. I just try to give you a little bit of rhyme or reason why they're put there, and what some of these terms mean, because hopefully that will help you organize that. But it's just a lot of kind of memorizing what's in what division and what it does.
Do you have any like flow chart that you could show us, or that's something we have to like make on our own?
It's something that I think you're better off making on your own, because it will stick with you in your mind more.
Okay.
I'll try to find, one that I can share with you. The reason I say it's better to make it on your own is because you'll probably make connections that mean more to you, right?
Mm-hmm.
The way I might make a flow chart might be a little bit different than Amy or Mary or Katie, or so that's the best reason for it.
Okay, thank you.
You're welcome. But I want to ask you to do those sorts of things as homework, and not because it's busy work, although it might seem like busy work at the time, but because it's going to help you and benefit you when it comes to the test, right? It's going to be a study guide that you can use for your test. So I did record this. I will post this like I did with the last one. Are there any other questions before I let you guys go?
I have a question.
Sure.
My question is, when are we going to submit the homework?
You should have submitted it already. If you go to Blackboard and you go to the particular homework assignment, there should be a place to upload the assignment right there.
So I'm
Just
late?
click on. Yes, you're late, and I'll give you credit for it. I'm not going to, I mean, it's only the first homework assignment, so everybody's kind of getting used to this, and I understand that.
Are you,
Right.
are you posting the next assignment?
I will post the next homework for chapters 11 and 12 tonight,
Okay, thank
you
you.
know. Yeah, you're welcome. And in general, I'm giving you guys about a week to do the homework, because I want you to keep moving to the next chapters, right? So I don't want you to get bogged up
Yeah.
and end up having 4 homework assignments due.
Yep. Yep.
Any questions? Right. Well, you guys have a great weekend. Like I said, I'll post the homework assignments tonight, and I'll start grading your homeworks from last week as well tonight, so that you can get some feedback on those.
Thank you. Bye,
You're
Rick.
welcome.
Thank you. Have a good week.